Vienna, 1902. A cramped laboratory in the Pathological Anatomical Institute. Two researchers, Adriano Sturli and Alfred von de Castello, are bent over a microscope, staring at a blood sample that makes absolutely no sense. For the past year, their mentor Karl Landsteiner has been mixing blood from different people in test tubes, watching red cells clump and separate in distinct patterns. He's proven that human blood falls into three groups, A, B, and a third he called C, later renamed O. Each group carries specific markers on its red blood cells, antigens that trigger immune reactions when mixed with the wrong blood. It's elegant, it's predictable, it explains centuries of mysterious transfusion deaths. But this sample on their slide, it's doing something impossible. It carries both A and B antigens on its red cells as if it belongs to two groups at once, and its serum, the liquid part, contains no antibodies against either. No anti-A, no anti-B, nothing. By the rules Landsteiner just discovered, this blood shouldn't work. It should attack itself, trigger a catastrophic immune reaction, kill its owner before birth. But here it is, drawn from a living, breathing person, defying every pattern they've established. Sturli and von de Castello publish a short paper describing this fourth type. They don't name it yet, they barely understand it, but they've stumbled onto one of the strangest quirks in human biology. Blood type AB, the newest bloodline, the rarest group. The blood that, by the cold logic of immunology, arguably shouldn't exist at all. Today, roughly 4 to 6% of all humans carry AB blood. In some populations, especially across parts of East Asia, it's slightly more common. In others, particularly among indigenous peoples of the Americas, it's virtually absent. And among all blood types, AB holds a unique paradox. Because it carries both A and B antigens, people with AB blood can receive red cells from any ABO group, making them universal recipients. But because their plasma contains no anti-A or anti-B antibodies, that same plasma can be given safely to anyone, making them universal plasma donors. Simultaneously the most compatible and the most restricted, the most forgiving and the most specific, a blood type that breaks its own rules. If you're listening to this to fall asleep, you're in the right place. That's exactly why I make these. So maybe subscribe, and if you feel like it, drop a comment telling me where you are and what time it is. I love seeing Berlin at breakfast, Boston at bedtime, Tokyo at dawn, all in the same thread. It's like a quiet map of people falling asleep to the same strange stories at different hours. This is the story of that blood, how it arrived last in the lab but carries genes older than our species, how it emerged from the meeting of ancient human migrations, written into existence when populations that should never have mixed anyway. How plagues and cholera and COVID-19 shaped who carries it and who doesn't. How it became tangled in eugenics, personality myths, and the dark history of categorizing people by what flows in their veins, and what it remembers, invisibly, about where we came from. The newest bloodline, the oldest story. But let's go back. Back before AB was identified, before anyone knew blood came in types at all. Back to a world where transfusion was less medicine and more desperate gamble, where doctors poured blood from one body into another with no idea whether they were saving a life or killing someone in the most agonizing way imaginable. 1667. Paris. A young physician named Jean-Baptiste Denis is about to make history. He's read about William Harvey's discovery of blood circulation, seen the experiments in England where Richard Lower kept dogs alive by transfusing blood between them. If it works in animals, Denis reasons, why not humans? On June 15th, he takes a 15-year-old boy who's been sick with fever, repeatedly bled by other doctors until he's weak and pale, and he transfuses about 12 ounces of lamb's blood directly into the boy's veins. And the boy lives, not only lives, but reportedly feels better. Word spreads fast. Dennis becomes famous overnight. Patients begin seeking him out, desperate people willing to try anything. But then the pattern starts to break. A Swedish nobleman receives sheep's blood and survives. Another patient isn't so lucky. Antoine Moroy, described as mentally disturbed and violent, receives multiple transfusions of calf's blood over several weeks. After the third transfusion, he develops a raging fever. His urine turns dark, almost black. He collapses. Within days, he's dead. Dennis is brought to trial in 1668, accused of killing Moroy. He's eventually acquitted. The court suggests Moroy's wife may have poisoned him, but the damage is done. Transfusion, the court declares, can only proceed with approval from the Faculty of Medicine. Effectively, it's banned. Across the Channel, Richard Lower and Edmund King are conducting their own experiments before the Royal Society. They transfuse sheep's blood into a volunteer, Arthur Koga, in November 1667. Koga survives, even claims to feel improved. But the theological objections are mounting. Blood, many argue, carries the soul, the essence of the creature. Mixing animal blood with human blood is unnatural, possibly blasphemous. By 1670, France has formally prohibited human transfusion. England follows. Italy joins. For nearly 150 years, the procedure vanishes from mainstream medicine, driven underground by a mystery no one can solve. Sometimes it works, sometimes it kills, and there's no way to predict which. Then in the 1820s, a London obstetrician named James Blundell decides to try again. He's watched too many women bleed to death after childbirth, hemorrhaging faster than their bodies can replace the lost volume. Animal blood is clearly the wrong answer, he reasons. But what about human-to-human transfusion? In 1818, he performs the first documented successful human-to-human transfusion. Over the next decade, he reports about 10 cases. Half seem to work, women pulled back from the edge of death, their colour returning, their pulse strengthening. The other half go terribly wrong. Patients develop sudden fevers, crushing chest pain, dark urine, organ failure, death. Blundell keeps meticulous records, but he can't find the pattern. Same technique, same volumes, completely different outcomes. Other physicians pick up where Blundell left off. Throughout the 19th century, they document case after case, building an increasingly detailed picture of transfusion reactions without understanding what they're looking at. Patients who survive describe feeling warmth spreading through their bodies, a sense of strength returning. Patients who don't survive describe something else entirely, a feeling of impending doom, pain radiating from the chest and back, skin flushing, then turning grey, and that dark urine, tea-coloured or nearly black. The signature of something catastrophic happening inside. We know now what they were seeing. Acute hemolytic transfusion reactions. When incompatible blood enters the body, antibodies in the recipient's plasma attack the foreign red cells, ripping them apart. Hemoglobin spills into the bloodstream, overwhelms the kidneys, triggers cascading organ failure. But in the 19th century, none of this made sense. Blood was blood. The idea that it came in types, that invisible markers on red cells and invisible antibodies in plasma were deciding life and death, that concept didn't exist yet. Doctors were operating blind, watching patients die from a variable they couldn't see, measure, or predict. The invisible war was happening on the surface of every red blood cell, written in antigens and antibodies in the language of A and B and O, but nobody could read it yet. Not until Karl Landsteiner walked into his Vienna laboratory and decided to mix blood in test tubes instead of bodies, to watch the reactions happen under glass instead of inside dying patients. That's when everything changed. That's when the letters finally appeared. Vienna, 1900. The Habsburg Empire is at its twilight. Mahler is conducting at the Opera House. Freud is publishing his theories about dreams. And in a laboratory at the Pathological Anatomical Institute of the University of Vienna, a 32-year-old pathologist named Karl Landsteiner is doing something that seems almost absurdly simple. He's mixing blood samples in test tubes, not in bodies, not in desperate operating theatres with dying patients and no way to predict the outcome, just in glass tubes on a laboratory bench, methodically combining red blood cells from one person with blood serum from another, over and over, watching what happens under conditions where he could actually see the reactions unfold. Landsteiner had spent years conducting autopsies, examining the bodies of patients who died from infections, from mysterious fevers, from complications no one could explain. He'd developed an almost obsessive attention to detail, a conviction that if you looked carefully enough at biological systems, patterns would emerge from the chaos. So he recruited volunteers, colleagues from the institute, laboratory assistants, himself. He drew blood from each of them and separated it into components. Red cells in one tube, the clear yellowish serum in another. Then he started mixing. Red cells from person A with serum from person B. Red cells from B with serum from C. Every possible combination, carefully labelled, systematically observed. And what he saw changed everything. Sometimes the mixtures remained smooth, red cells floating individually in the liquid. But other times something dramatic happened. The cells would clump together, forming visible aggregates that settled to the bottom of the tube like red snow. Agglutination, it was called. The same reaction doctors had been seeing inside dying transfusion patients for 200 years, now visible on a laboratory bench where it could be studied, quantified, understood. At first it seemed random, but Landsteiner was meticulous. He tested dozens of combinations, made careful notes, looked for patterns in the chaos, and gradually the pattern revealed itself. The samples weren't mixing randomly. They fell into distinct groups based on how they reacted with each other. There were people whose red cells were agglutinated by some sera but not others. People whose serum caused agglutination in some samples but left others untouched. It was like a chemical conversation happening on the surface of every red blood cell, invisible to the eye but absolutely consistent in its logic. In 1901, Landsteiner published his findings in a paper that was remarkable mostly for how unremarkable it seemed at the time. No grand declarations, no revolutionary claims, just a careful description of what he'd observed. He'd identified 3 distinct patterns, which he labelled A, B, and C. Group A red cells reacted with the serum from Group B individuals, but not with A serum. Group B cells showed the opposite pattern, clumping with a serum but not B. Group C cells, the ones he'd find would later be renamed O, were agglutinated by both A and B sera. 3 groups, 3 distinct chemical identities, 3 invisible tribes that every human being belonged to whether they knew it or not. The paper attracted almost no attention. A few colleagues took note. Most of the medical establishment ignored it entirely. What difference did it make that blood could be divided into categories in a test tube? Transfusion was still dangerous, still unpredictable. Landsteiner had found a pattern, but he hadn't yet explained what it meant or how to use it. Then, in 1902, something unexpected happened. 2 of Landsteiner's colleagues at the Vienna Institute, Adriano Sturli and Alfred von de Castello, were continuing his work, testing more blood samples, looking for exceptions to the 3-group model, and they found one. Actually, they found 2. 2 donors whose blood didn't fit any of Landsteiner's categories. Their red cells carried both A and B antigens, something that shouldn't have been possible according to the neat 3-group scheme. And their serum, instead of containing antibodies against A or B or both, contained neither. It was a fourth pattern, a combination type that broke the rules. They called it AB. For Landsteiner, it must have been both frustrating and fascinating. Just when the system seemed to make sense, here was an exception that complicated everything. But the exception was revealing something profound about how blood groups actually worked. At the molecular level, though Landsteiner couldn't have known this in 1902, blood types are determined by enzymes that add specific sugar molecules to the surface of red blood cells. The A enzyme adds one type of sugar called N-acetylgalactosamine. The B enzyme adds a different sugar called galactose. Both enzymes modify the same base structure, the H antigen turning it into A or B. People with O blood have a non-functional enzyme, so their red cells carry only the unmodified H antigen. But people with AB blood have both working enzymes. Both sugars get added. Every red cell in their body displays both A and B antigens side by side. And here's the immunological paradox that makes AB so strange. Normally, your immune system produces antibody against any blood group antigens you don't have. If you're type A, you make anti-B antibodies. If you're type B, you make anti-A. If you're type O, you make both. Your immune system learns early in life what belongs on your red cells and produces antibodies against everything else. But AB individuals have both antigens from birth. Their immune system sees both A and B as self, as part of the body's normal landscape, so it produces neither anti-A nor anti-B antibodies. They're the only blood type that doesn't generate natural antibodies against other human blood groups. This makes AB blood immunologically unique in two contradictory ways. For red cell transfusions, AB is the most restricted recipient. An AB patient can only safely receive AB blood because their red cells carry both antigens and would be attacked by the anti-A or anti-B antibodies present in AB or O donor blood. But for plasma transfusions, AB is universal. AB plasma contains no anti-A or anti-B antibodies, so it can be given to anyone without triggering a reaction. Two sides of the same immunological coin. Universal plasma donor, restricted blood recipient. A blood type that works by the rules while simultaneously breaking them. Landsteiner's work, and the discovery of AB that followed, should have revolutionized medicine immediately. It didn't. The paper was published in a relatively obscure journal. The implications took years to sink in. Doctors continued performing transfusions the old way, mixing blood types at random, watching some patients live and others die. Slowly, gradually, a few pioneering surgeons began testing donors and recipients before transfusion, matching blood types according to Landsteiner's scheme, and suddenly, transfusion stopped being a lottery. Matched patients survived. Mismatched patients had reactions. The mystery that had haunted medicine for 250 years dissolved the moment someone thought to look at blood under controlled conditions instead of crossing their fingers and hoping. Landsteiner moved to the Rockefeller Institute in New York in 1922, continued his research into blood and immunity, discovered the Rh factor that would further refine transfusion safety. But it took until 1930 before the medical establishment fully recognized what he'd done. That year, nearly 3 decades after his original discovery, Carl Landsteiner was awarded the Nobel Prize in Physiology or Medicine for discovering human blood groups. The citation was simple, elegant. He'd shown that blood wasn't one thing but many, that invisible markers divided humanity into immunological tribes, and that understanding those divisions could save lives. And AB, that fourth pattern discovered almost as an afterthought by his colleagues, remained the rarest and strangest of the groups. A blood type that combined what shouldn't combine, that existed at the intersection of two ancient genetic lineages. The newest bloodline in the oldest story. The type that shouldn't work but somehow does. Here's the puzzle Landsteiner handed to immunology. Take any person with type A blood and examine their plasma under controlled conditions. You'll find antibodies against B antigens floating in their bloodstream. They've never been transfused. They've never encountered B-positive red blood cells in their life. Yet their immune system is already armed and ready to attack them. The same pattern holds for type B individuals. Their plasma contains anti-A antibodies, preformed and waiting, targeting an enemy they've never met. Type O individuals produce both anti-A and anti-B, a double arsenal of antibodies ready to destroy any red cells carrying either antigen. This is Landsteiner's rule, the foundation of transfusion safety, and one of the most reliable patterns in human immunology. Your body makes antibodies against the blood group antigens you lack. Always, except when it doesn't. Those antibodies appear in infancy, weeks or months after birth, without any obvious trigger. For decades, this seemed mysterious. How could babies produce antibodies against blood types they'd never encountered? The answer, when it finally emerged from studies of gut bacteria and environmental microbes, was almost poetic. The sugars that define A and B antigens aren't unique to human blood. They show up everywhere in nature, on the surface of bacteria in your intestines, on pollen grains in the food you eat. As an infant's immune system develops, it encounters these bacterial A-like and B-like sugars constantly. If those sugars don't match what's on your own red blood cells, your immune system learns to recognize them as foreign, potentially dangerous. It starts producing antibodies. By the time you're 6 months old, your blood contains a standing army of anti-A or anti-B antibodies, depending on your type, ready to attack transfused red cells, ready to destroy incompatible blood before it can circulate, ready to kill you if a doctor makes the wrong choice in an emergency. This is elegant defensive immunology. Your body surveys the landscape of sugars on your own cells, compares them to sugars it encounters in the environment, and produces antibodies against anything that doesn't match. It's the same logic that protects you from bacterial infections and viral invasions. Recognize self. Attack everything else. Simple, effective, universal, except in type AB, because AB individuals have both A and B antigens on their red blood cells from birth. Both patterns are recognized as self from the moment the immune system begins learning what belongs in the body. So when AB infants encounter A like sugars on bacteria in their gut, their immune system sees them and thinks, that's normal, that's me. When they encounter B like sugars, same response. That's self. That's safe. No antibodies form. By adulthood, AB plasma is essentially clean of anti-A and anti-B antibodies, not because the immune system failed, because it succeeded perfectly at recognizing both antigens as self. But this creates an immunological paradox that still puzzles researchers. From the perspective of immune defense, AB should be the worst possible configuration. You're carrying two major antigen families that other blood types recognize as foreign. You're displaying both flags simultaneously, and you've disarmed yourself, producing neither of the antibodies that would protect against A or B contamination. In a world where blood types mattered for survival, where incompatible blood transfusions happened naturally through some mechanism we don't understand yet, AB should be an evolutionary disaster, yet here it is, rare but persistent, present in every major human population, somehow stable across thousands of generations, and the clinical implications are a mirror image paradox. For red blood cell transfusions, AB is the most restricted recipient type. An AB patient needs AB blood. Anything else brings antibodies that will attack their transfused cells, or brings red cells lacking the antigens the patient's system expects. In practical terms, this makes AB patients dependent on the rarest compatible donor pool, only about 4% of most populations. Emergency situations where AB blood isn't available become complicated. require careful cross-matching, carry higher risk. But flip the equation. Look at plasma instead of cells, and AB becomes the universal donor. AB plasma, free of anti-A and anti-B antibodies, can be transfused into anyone without triggering an immune reaction. It's liquid tolerance, a substance that won't attack A red cells or B red cells or O cells because it contains none of the weapons that cause transfusion reactions. Blood banks prize plasma for exactly this reason. For emergency transfusions when there's no time to type and match, for massive trauma cases requiring volume replacement fast, AB plasma is universal, the safest choice. Now compare that to type O. O red cells are universal donors because they lack both A and B antigens. No targets for anti-A or anti-B antibodies to attack. You can give O negative red cells to almost anyone in an emergency and expect them to survive. But O plasma is the opposite of universal. It's loaded with both anti-A and anti-B antibodies, the most aggressive antibody cocktail of any blood type. Transfuse O plasma into an AB or AB patient, and those antibodies will attack their red cells immediately. The same blood type that's safest for red cell transfusion is the most dangerous for plasma, and the same type that's most restricted for red cells, AB, is the safest for plasma. Two perfect inversions, two mirror image paradoxes in the same biological system. Evolutionary biology doesn't have a neat answer for why AB exists. The textbook explanation is balancing selection, the idea that maintaining multiple blood types in a population provides advantages that outweigh the costs. Maybe different types resist different infections. Maybe some configurations improve pregnancy outcomes, while others enhance clotting. Maybe the variation itself is valuable, ensuring that no single pathogen can exploit one immunological weakness to devastate an entire population. Under this model, AB persists not because it's advantageous, but because the A and B alleles that create it are maintained by selection, and AB is simply what happens when both show up in the same person, a byproduct of diversity, a side effect of variation that's protective at the population level even if it's puzzling at the individual level. But there's another possibility, one that's harder to test but more intriguing. Maybe AB's immunological openness isn't a bug. Maybe it's a feature. Maybe having a blood type that doesn't mount antibody responses against A or B antigens provides subtle benefits we haven't identified yet. Benefits in pregnancy compatibility, in autoimmune regulation, in responses to certain infections or inflammatory conditions. The data is scattered, contradictory, never quite definitive. But the fact that AB exists at all, that it hasn't been eliminated despite seeming to violate basic immune logic, suggests that somewhere in our evolutionary past tolerance had value, even when defense seemed more important. To understand why tolerance might matter, you have to step back, not hundreds of years to the first blood transfusions, not thousands of years to the first cities, millions of years, back before humans, back before Neanderthals, before Denisovans, before anything we would recognize as our own kind. Because the ABO system is not a human invention. It's not even a human trait in the narrow sense. It's older than our species, older than our genus, older than our branch of the primate family tree. Inside every one of your cells, on chromosome 9, there is a stretch of DNA at position 9q34.2. Molecular biologists call it the ABO locus. It's a recipe for an enzyme, a glycosyl transferase, whose job is strangely humble. It takes a sugar molecule and attaches it to a small scaffold on the surface of your red blood cells, something called the H antigen. That little addition changes everything. Different sugars, different blood types. In type A, the enzyme adds one particular sugar, N-acetyl-galactosamine. In type B, a nearly identical enzyme adds galactose instead. On paper, the difference between those enzymes is tiny. 4 amino acids changed out of hundreds. 4 microscopic switches in a protein chain, and suddenly your blood belongs to a different immunological tribe. For most of the 20th century, it was tempting to think those switches had flipped recently. A quirk of human evolution. Maybe some ancient mutation in Africa. Some tribal founder effect. Some small local story. Then geneticists started sequencing the same region in other primates. Chimpanzees, gorillas, gibbons. They found something unnerving. They found A. They found B. Not just vaguely similar versions, not just a fuzzy echo. They found the same basic structure, the same key amino acid positions, tuned to the same 2 solutions. An A-like enzyme that adds 1 sugar, and a B-like enzyme that adds another. The same immunological dialect spoken in different primate languages. In some chimpanzee populations there are individuals whose red cells react like human type A. Others like type O. In gorillas there are B-like variants. Gibbons carry their own mix of A-like and B-like alleles. The details differ, the frequencies shift, but the pattern is unmistakable. The A/B divide is not exclusive to us. That raises a profound question. Did each species independently evolve its own version of A and B, converging on the same biochemical solution again and again? Or are we all sharing something more ancient, a genetic relic from a common ancestor deep in the primate past? Modern population genetics leans toward the second answer. The A and B lineages at the ABO gene look old, older than the split between humans and chimpanzees, older than the split between great apes and lesser apes, possibly older than the split between major primate groups altogether, tens of millions of years old. Geneticists have a name for this pattern, transspecies polymorphism, a variation that doesn't respect species boundaries, that survives speciation events, passing from ancestor to descendant as the tree of life branches. Most genetic variants die out or fix within a few million years, but some, under the right kind of selection, are preserved across whole radiations of species like a refrain in a long, complicated song. A BO seems to be one of those refrains. What this means, if the models are right, is quietly astonishing. When you talk about A and B blood group alleles, you're not talking about something that arose in human prehistory. You're talking about a molecular distinction that your ancestors shared with the ancestors of chimpanzees, gorillas and gibbons, long before there were humans, long before any hominin walked upright across an African savanna. Your blood is carrying a decision evolution made in some forgotten Eocene forest, in a world of different climates and different predators and different pathogens. Why did that decision persist? The best guess is balancing selection. Something in the environment, probably infectious disease, kept both A-like and B-like enzymes valuable over very long timescales. Maybe one configuration protected better against one parasite, the other against a different microbe. Maybe pathogens themselves evolved to mimic one sugar but not the other, pushing populations to keep both options in play so no single microbe could exploit everyone. Whatever the exact pressures were, they were strong enough and stable enough that natural selection did something it almost never does. It held onto 2 rival solutions through wave after wave of extinction and speciation, preserving the same basic A/B contrast in multiple primate lineages for millions upon millions of years. Underneath all the modern labels, A positive, B negative, ABO, this is what you are. A primate carrying an ancient immunological code that predates your species by an order of magnitude. But not every piece of the ABO puzzle is that ancient. The human O allele, the one that produces type O blood, tells a different kind of story. If you read its DNA sequence carefully, you find a glitch, a single-letter deletion in one of the crucial exons. That tiny deletion pushes the reading frame of the gene out of alignment. The resulting enzyme is truncated, misfolded, basically useless. It never gets the chance to add a sugar to the H antigen. the underlying scaffold remains bare. To your immune system, that blankness becomes its own identity. No A sugar, no B sugar, type O. B. From a biochemical perspective, O is not a third elaborate design, not a parallel to A and B. It's a loss of function, a broken tool, a gene that used to do something and now, in this particular variant, no longer does. And it's not even a single broken tool. There are many O alleles in humans, different deletions, different mutations that all converge on the same end result. No working enzyme, no added sugar. That pattern suggests something surprisingly counterintuitive. O hasn't just happened once. It has happened over and over again, independently, in different human lineages. Where A and B seem to be the ancient, carefully maintained pillars of the system, O looks like a more recent improvisation, a series of accidents that turned out to be useful enough to keep. So you have this layered history folded into one gene. At the foundation, two old, deeply conserved functional alleles, A and B, shared across species, moulded by millions of years of pathogen warfare. Stacked on top of that, in our particular branch, a collection of newer disabling mutations that give rise to O. And somewhere in that space of possibilities, tucked between the ancient and the recent, sits AB. Genetically, AB is simple. It is not a special allele of its own. There is no AB gene waiting in your genome. An AB person is simply someone who inherited a working A allele from one parent and a working B allele from the other. Codominance does the rest. Both enzymes are made. Both sugars are attached. Both antigens appear on the red blood cell surface side by side. From the perspective of deep time, that combination could have happened whenever A-carrying and B-carrying lineages mixed. An A-like primate from one group mates with a B-like primate from another, and suddenly, in that offspring, both surface sugars are present at once. Some version of that must have occurred many times in many species, but in humans, AB does not feel like a simple recombination. It feels like a historical event, a meeting. Because unlike the alleles themselves, which are old, the pattern of how those alleles are distributed is recent. Human populations are not uniform. Some regions of the world have long been dominated by A, others by B, others by O. Geography, migration, drift, and disease all shape those frequencies differently in different places. The AB phenotype depends on more than just the existence of A and B alleles in the species. It depends on population dynamics. It depends on people moving, settling, trading, marrying outside their birth groups. It depends on formerly separate streams of ancestry crossing and blending until children begin to appear carrying one ancient allele from each side. That is why AB feels new. The ingredients are ancient. The recipe is not. For most of human evolution, our ancestors lived in relatively small, somewhat isolated populations, shaped by local pathogens and local histories. A-heavy groups could go generations without significant contact with B-heavy groups. In those conditions, AB would exist, rare, scattered, a byproduct of occasional mixing, but it would not define whole communities. It would not form a visible bloodline in the way we think about it now. For AB to become more than a statistical whisper, the world had to change. Populations had to move farther and faster. Trade routes had to knit distant regions together. Empires, migrations, and diasporas had to take people whose ancestors had lived with one blood group profile for thousands of years and bring them face to face with people shaped by a different profile. Then they had to do the most human thing of all. They had to have children. When A met B, again and again, across centuries and continents, AB stopped being a rare accident and became a stable, if still uncommon, presence in the human story, a visible signature of mixed histories, layered ancestries, and long-distance connections. But that part of the story doesn't belong to deep time anymore. It belongs to us, to the last 10 or 15,000 years of settled life, trade, and conquest. to the Silk Road, to steppe migrations, to ports and caravanserais and borderlands where strangers shared food, stories and genes. To see how the newest bloodline finally emerged from this web of ancient alleles and human journeys, you have to follow those movements. You have to watch A-dominated populations and B-dominated populations collide, cooperate and intermarry. You have to watch what happened when A met B. If you could drain the whole planet of colour and just leave one thing glowing, imagine lighting up only the A alleles. Not people, not flags, not borders. Just that one ancient variant, humming quietly inside red cells. You'd see a strange map. Across much of Western and Northern Europe, a soft, steady brightness. Scandinavia, the British Isles, down through parts of Central Europe. Cross the Arctic and you'd find it again in small, cold communities of northern North America and Greenland. Far away in the southern hemisphere, patches of light would burn in Aboriginal Australia, in groups where A and O dominate and B is almost absent. Scattered points would appear in some native North American nations, little islands of A in a wider sea. Now dim that and turn on B. The map shifts east and south. Central Asia begins to glow. The great steppe belt, the lands that would one day carry horse archers and caravans, light up with B. India brightens from the Indus plain across to the Ganges basin and beyond, a dense, shimmering field. Northern China, parts of Eastern Europe, arcs of Central and South Asia. Here B is strong. In contrast, Western Europe fades to a faint dullness. Most of Australia goes dark. Large stretches of the Americas remain almost black, at least in their pre-colonial state. Two old lineages, same species, very different geographies. O is everywhere, threaded through almost all of these regions in varying degrees, a quiet background presence. But if you only watch A and B, you begin to see something else. You start to see edges, frontiers, zones where the two patterns press up against each other. Those edges are where AB lives, because AB is not a free-floating thing. It's a coincidence, an overlap. To have AB, someone in your ancestry had to carry A, someone else had to carry B. At some point they had to meet. In places where almost everyone is A and O, with almost no B at all, AB barely appears. It can't. In places where nearly everyone is B and O, but it never really took hold, the story's the same. But in the long, irregular band where A-heavy and B-heavy populations have brushed against each other for centuries, something different happens. Contact zones. Geneticists sometimes talk about clines and gradients. Anthropologists talk about frontiers and corridors. In the language of this story, you can think of them as long, thin rooms where history has kept leaving the door open. Central Asia is one of those rooms. The steppe and its fringes, from Eastern Europe through Kazakhstan and beyond, have seen waves of movement for tens of thousands of years. Hunters following herds, pastoralists following grass, traders following profit. Here, A from the west and B from the east have had a very long time to swirl together. Modern blood surveys hint at the result. In several Central Asian populations, both A and B are relatively common, and AB rides higher than the world average, reaching levels that would look unusual in much of Europe or Africa. It's as if the old lines have blurred just enough that the hybrid combination begins to show up again and again. Follow those corridors east, and the pattern intensifies. Across parts of East and South Asia, you find some of the highest AB frequencies on Earth. Bangladesh, with estimates around the high teens for AB positive. Pakistan, India, Nepal. Multiple surveys suggesting roughly 1 person in 10 may carry both antigens. In Kazakhstan and its neighbours, AB rises again, a quiet testament to long histories of mixing in the heart of Eurasia. And then there is the archipelago of high AB that has fascinated demographers for decades, Japan and the Korean Peninsula. If you walk through the numbers slowly, the way you might trace a coastline, you find a distinct profile. In Japan, large studies report something like 39% A, just over 20% B, about 30% O, and roughly 10% AB. South and North Korea show similarly elevated AB, often around 9 to 11%. These figures shift a little depending on the survey, the year, the sample. They are not precise coordinates, but they tell a consistent story. Japan and Korea are places where A and B are both strong. When both parents' lineages are rich in A and rich in B, AB stops being a rare accident and becomes a stable minority. Not common exactly, but no longer surprising. In a classroom of 40 students in Tokyo or Seoul, there might be 4 AB children. In many European schools there might be 1. In some indigenous American groups, historically, there might have been none at all. How did that happen? No one can point to a single migration or a single war and say, that's when AB began. The data are not that sharp. What we have instead are overlapping, sometimes competing pictures. Archaeology, linguistics, and genetics all suggest that East Asia has been a crossroads for a very long time. Early farming cultures spreading from the Yellow River Basin, older hunter-gatherer lineages along coasts and islands, later movements from the north and west, all layering over each other. On the Korean peninsula, successive waves from mainland Asia met older populations and over centuries blended. Across the narrow straits, the Japanese islands saw their own sequence of arrivals. Jomon hunter-gatherers with deep roots, then Yayoi agriculturalists carrying rice, metal and new genes from the continent, and later historical contacts with Korea and China. Each encounter, each period of peaceful intermarriage or forced assimilation would have brought different ABO profiles together. If A was already relatively well established in some of those lineages and B more common in others, then the long, slow churn of East Asian history would have done the rest. Marriages across clan boundaries, political unions, captives absorbed into new communities, merchant families settled in foreign ports. Over many generations, the old A-rich and B-rich strands would interweave until the children of the region often carried both. From that perspective, the high AB frequencies in Japan and Korea are not a mystery at all. They are what you would expect in a place where diverse ancestries have been recombining for a very long time, and where both A and B have, by chance and selection, found comfortable niches. Farther west, in the great band of land once tied together by the Silk Road, a similar but not identical process may have played out. Picture the old caravan routes by night, desert stations where Central Asian herders met Persian traders and Chinese envoys, oasis towns where Indian merchants unloaded spices and textiles next to steppe nomads selling horses, inns and caravanserais where people who had grown up 1,000 miles apart shared fires, stories, and occasionally beds. From a genetic point of view, the Silk Road was not a single road at all, but a mesh, a network of movement running from the Mediterranean shores through Central Asia to the Pacific coast, with side branches south into India and north into Siberia. Over centuries it moved more than silk and spices. It moved people, soldiers, slaves, artisans, brides. The Mongol Empire, when it rose, supercharged that process. In the 13th century, mounted armies from the heart of the steppe swept east into China, west into Eastern Europe, south into the Islamic world. They burned cities, toppled kingdoms, and then, paradoxically, created one of the largest contiguous zones of relatively safe long-distance trade in history. Merchants could, for a brief span of time, cross thousands of kilometers under imperial protection. It's tempting to imagine those centuries as a kind of great shuffle of alleles, A biased populations in the west, B biased populations in the east, drawn into closer and more sustained contact, their descendants mixing in border towns, garrison cities, and conquered capitals. over generations that could push AB from a thin statistical whisper to a more audible note in the human chorus. But this is where caution is important. We do not have a fossil record of blood types. No ancient skeleton has a label that says AB positive 1250 CE, born near Samarkand. When researchers try to reconstruct the past of ABO frequencies, they are working backwards from modern data, from a few ancient DNA samples where preservation allows, and from models of how alleles drift and spread in populations. These are powerful tools, but they give ranges and probabilities, not exact histories. So when we say the Silk Road and the Mongol Empire may have increased AB frequency in Eurasia, that may is doing real work. It marks the line between what is strongly suggested by patterns and what is directly observed. What is clear is broader and in some ways more poetic. A B follows the seams. Where human groups with different ABO profiles have remained isolated for millennia, like many pre-Columbian populations in the Americas, heavily dominated by O, or some Aboriginal Australian groups where B is almost absent, AB stays vanishingly rare or essentially non-existent. Where those groups have met, mingled and settled together, Central and South Asia, parts of East Asia, old crossroads and migration corridors, AB rises. It's not that AB is a mixed-race blood in any crude or modern sense. Human variation doesn't sort itself neatly into our political or social categories. But at a molecular level, an AB person really is the meeting point of two very old lineages, one parent or one ancestral line carrying A, another carrying B, both ultimately descending from decisions evolution made in some ancient primate forest, but travelling through different human histories before they finally converged. Look at it that way, and an AB individual becomes a kind of living document. Their red cells are recording without words that somewhere in their family tree a boundary was crossed. Maybe it was a literal frontier between tribes, or a social line between castes, or a gradual blending along a river valley where traders married locals and never left. Maybe it happened 5 generations ago, maybe 50. The dates are lost. The signature remains. This is why AB feels both new and old at once. Old, because the A and B alleles themselves are ancient, shared across primates, maintained by millions of years of disease and selection. New, because the particular human patterns that bring those alleles together in large numbers are tied to recent history, to the spread of agriculture, to the rise and fall of empires, to boats and horses and long roads. New too in the sense that AB is still rare. Globally, perhaps only 4 to 6 people out of 100 carry it. In some countries, fewer. In others, like Japan and Korea, or parts of South Asia and Central Asia, closer to 10 or more. Those numbers shift with migration, with birth and death, with the quiet arithmetic of population genetics. They are not fixed, but behind them is a simple rule. Where A travels and B does not, AB will be scarce. Where B travels and A lags behind, AB will be scarce. Where both travel, settle, and stay, generation after generation, AB begins to accumulate. It is the overlap made visible. If your blood is AB, your body is carrying that overlap. Somewhere in you, 2 lines of descent that once walked very different paths have been braided together. You are quite literally where A met B, and that, in turn, has mattered for more than just how your blood behaves in a hospital. Because while human movement and mixing brought A and B together, something else was always there in the background, shaping who survived long enough to pass those alleles on. Disease, plagues, parasites, and pandemics have been quietly editing the ABO map for as long as humans have been human. And AB, born from the meeting of 2 ancestral strategies, carries their fingerprints too. If you zoomed out from the map of migrations and trade routes, and instead watched time itself, speeding centuries past like clouds, you'd notice something unsettling. Wherever humans gathered in numbers, something else gathered too. First as small sparks in the record, odd clusters of burials, strange lesions on ancient bones, then as waves described in chronicles and temple inscriptions, fevers, swellings, rashes, sudden death. The language changes. The gods blamed for it change, but the pattern is the same. Everywhere people go, diseases follow, and they do not follow randomly. We tend to think of blood types as a hospital problem, a matter of transfusions and donor cards. But long before anyone had a word for Group A or Group B, microbes were already reading those invisible letters, probing them, exploiting them, being blocked by them. Because those A and B signatures are not just on red cells, they're sugars, tiny molecular decorations, posted like signboards on the surfaces of many of your cells, especially in your gut. To a bacterium or a parasite, those sugars are not abstract categories, they're footholds, or obstacles, or food. Over generations, that relationship becomes a negotiation. Pathogens testing every door. Human bodies changing the locks. In genetics, this is called pathogen-driven selection, the slow, statistical favouring of whatever variants let you dodge the worst of an epidemic, even if they come with their own quiet costs. The ABO system is one of the clearest places where we can see that negotiation written down. Take cholera. In the crowded river deltas of South Asia, especially around the Ganges and Brahmaputra, Vibrio cholerae has been part of the background for a very long time. It lives in brackish waters, hitchhiking on plankton, waiting for a chance to explode into human populations when sanitation fails and floodwaters rise. Most infected people will never know they met it, a short illness, or none at all. But some are hit with something else entirely, a catastrophic torrent of fluid loss that can drain a human body to the edge of death in a single day. When researchers in Bangladesh started comparing those severe hospitalized cases with the general population, a pattern emerged. People with blood group O were overrepresented among the sickest. Those with AB were, more often than not, missing from the hospital beds. The difference seemed to live right there on the surface of intestinal cells. Cholera toxin latches on and hijacks your gut's ion pumps, turning absorption into secretion. For reasons that are still being explored, cells from type O individuals appear to respond more violently, the molecular equivalent of flinging open every tap at once. Types with A or B, and especially AB, seem to mount a less extreme reaction on average. The result is not simple immunity. An AB person can absolutely get cholera. But in the unforgiving arithmetic of an epidemic, if type O is slightly more likely to tip into life-threatening dehydration, and AB slightly less, that tiny shift in odds is enough. In a region where cholera has been endemic for centuries, generation after generation, it may gently push the population away from O and toward non-O types, especially toward B, which appears to be particularly favoured there. Look at modern blood surveys in parts of the Ganges Delta. O is lower than you might expect for a large old population. B is high. A B, the combined child of A and B, rides higher than the world average. It is tempting to see, in those numbers, the faint fingerprint of a waterborne killer that has been quietly culling type O lineages for a very long time. Travel west and south into the hot, humid zones where another parasite reigns, and the picture flips. Here, the great shaper is not a bacterium, but a single-celled organism that hides in mosquitoes. Plasmodium falciparum, the deadliest cause of human malaria. Falciparum has its own way of using the ABO system. When it infects a red blood cell, it pushes parasite proteins out onto the surface, turning the cell into something sticky. Those infected cells grab onto uninfected neighbours and to the walls of tiny blood vessels, forming clumps called rosettes. Big, stable rosettes can clog capillaries and starve tissues of oxygen. That's one of the pathways into severe disease. Coma, organ failure, death. The A and B sugars on red cell surfaces help build those rosettes. They act like extra bits of Velcro, giving the parasite more to hold on to. Group O cells, which lack both A and B, form smaller, weaker rosettes that break apart more easily. In hospital wards and field studies across sub-Saharan Africa, that difference shows up in the statistics. People with Group O are less likely to end up with the most severe forms of falciparum malaria. Not immune, not safe, but on average significantly less likely to be pushed over the cliff. Those with non-O blood, AB and yes AB, carry the extra sugars that help the parasite's clumps hold firm. In the complex tangle of genes and exposures that decides how a particular infection plays out, that one detail nudges them toward higher risk. When you stack centuries of intense malaria transmission on top of that tiny repeated nudge, the population starts to tilt. In many heavily affected African regions, O rises to form a large majority. A survives. B does too, though often at lower frequencies than in parts of South or Central Asia. A B, needing both A and B to meet, is correspondingly rare. The map here isn't drawn by caravans and seafaring empires. It's drawn by mosquitoes, by the nightly rise and fall of biting insects above swamps and irrigated fields, by a parasite that reads your red cell sugars like a lock reads a key. Other diseases have probably been lifting and lowering different blood groups in other corners of the world, though the evidence gets murkier the further back we look. When medieval chroniclers described the Black Death in Europe, the buboes, the sudden collapse, whole households gone in a week, they had no concept of Yersinia pestis, the bacterium that modern science has plucked from ancient teeth. They certainly had no idea that their own blood might make a difference. In the 20th century, some anthropologists tried to look back with new tools. Comparing rough ABO reconstructions from pre-plague and post-plague skeletal samples, a few claimed to see shifts, maybe a decline in O, maybe a rise in A or B. It was a tantalizing idea that the Black Death had not only emptied villages, but had subtly rewritten the genetic script of those who survived. Later work has urged caution. The methods for reading blood type from old bones are indirect. The sample sizes are small. Different cemeteries give different answers. If plague did nudge ABO frequencies, its touch is faint enough that we cannot yet trace it with confidence. Smallpox leaves a similarly ambiguous trail. Before its eradication in the 20th century, variola virus was one of humanity's most relentless killers. In some societies, nearly every person was infected at some point. Survivors were left scarred, blinded, or immune. The unlucky simply vanished. A handful of historical and serological studies have suggested that people with blood group A may have been more likely to develop severe smallpox than those with O or B. If that pattern was real and strong, then the endless churn of smallpox epidemics in Europe, Asia, and parts of Africa could have leaned populations a little away from A and toward other types. But here too the evidence is patchy. Old hospital records are incomplete. Confounders lurk. The exact mechanisms by which a virus might favour one ABO pattern over another are still being sketched. So we're left with a layered picture. In some cases, cholera and falciparum malaria above all, the links between blood type and disease risk are supported by both lab work and large population studies. In others, plague, smallpox, the connections are still hypotheses, interesting but unproven. And in all cases, individuals are not destiny. A type O person can survive cholera. An AB person can die of malaria. These are shifts in probabilities, not fixed fates. Where does that leave AB? Somewhere in the middle of a tug of war. As a phenotype, AB inherits both A and B antigens. That means, in a malaria zone, it likely shares the vulnerabilities of its parents. The parasite does not care that your A came from one lineage and your B from another. It only sees extra hooks to grab. In a cholera-ridden delta, though, the picture may be kinder. The same dual display of A and B sugars that hurts you in malaria country may soften the effect of cholera toxin on your gut, nudging you toward milder illness compared with your O-type neighbours. In such places, the very fact that AB lacks anti-A and anti-B antibodies, the feature that makes its plasma so useful in modern transfusion medicine, might also modulate certain immune reactions in ways we are only beginning to understand. And then there are all the other infections, past and present, that interact with ABO in smaller ways. Noroviruses that prefer one blood group sugars when they bind to the gut. Helicobacter bacteria in the stomach. Possibly viruses we've never sampled from ancient graves, long gone but not entirely forgotten, because they left faint imprints in who carries which alleles today. Add it all up, and AB looks less like a clean advantage or a clear disadvantage, and more like a gamble that is paid out locally. In a landscape dominated by malaria, it may be slightly punished. In one dominated by cholera, it may be a little sheltered. In places where neither is strong, other subtler pressures may matter more, or simple chance. That is the invisible hand at work. Not a conscious guide, but the combined force of countless epidemics, quietly turning the evolutionary crank, killing some carriers of certain alleles more often than others, letting a few more children with particular combinations survive to adulthood, have families, and shuffle the genetic deck again. A B exists because A and B met, but it persists because, in enough corners of the world, the balance of diseases, migrations, and marriages has never made it decisively wrong to be both at once. Which brings us closer to our own time, and to a more recent wave of infection that once again turned global attention to the tiny differences written in blood. When a new coronavirus stepped onto the world stage, the old question resurfaced in hospitals and labs. Do these silent letters, A, B, O, and their rare hybrid AB, still matter? And if they do, what exactly are they trying to tell us now? The first whispers came from one city. Early 2020. Winter air over Wuhan. Hospital corridors crowded with patients coughing, feverish, short of breath. Doctors there did what doctors always do in a new outbreak. They counted. Who is getting sick? How old are they? What other illnesses do they have? And because ABO typing is routine in many Chinese hospitals, they could ask one more question. What blood do they carry? When the numbers were tallied and compared against the blood types of the surrounding population, a faint pattern emerged. People with Group A seemed to be turning up among the infected and the severely ill more often than expected. People with Group O, a little less often. It was not dramatic, not a curse on A or a shield for O, just a quiet skew in the statistics, small but persistent enough to catch the eye of researchers already primed by old data from the first SARS outbreak in 2003, where Group O had also seemed very slightly less vulnerable. Within weeks, as the virus moved along air routes and motorways into Europe, other hospitals began to look. In Italy, in Spain, in France, in the dense wards of New York City, doctors and data scientists opened their electronic records and traced the same question. Again, in several early cohorts, the pattern echoed Wuhan's. A bit more A among the hospitalized, a bit more O among those who never tested positive despite strong exposure. A B, as usual, was harder to read. It was rare to begin with. In some studies it appeared slightly overrepresented among cases. In others it blended into the background noise. Soon there were headlines. Blood type may affect COVID risk. Simple, alarming. The kind of message that slips easily into the human tendency to sort the world into lucky and unlucky categories. The deeper story was more complicated and more interesting. When epidemiologists pulled thousands of cases and millions of controls into meta-analyses, the number settled into something modest. Compared with other groups, type O seemed to have a somewhat lower chance of infection, perhaps around 3/4 the odds of non-O. Types A and AB on average showed slightly higher odds of testing positive. But the differences were small. Not a wall between you and the virus, more like a thin gust of wind, nudging probabilities by a few percent, and not every study saw even that. Large datasets from blood donor registries in places like the United States and Iran sometimes found no meaningful connection at all between ABO type and whether a person caught the virus or developed severe disease. What mattered far more consistently were things we already knew: age, existing heart and lung problems, obesity, immune status, and later vaccination. Still, the patterns in those early reports demanded an explanation. Why would a respiratory virus care what sugars you carry on your red blood cells? Part of the answer lies in where those sugars really are. As with cholera and malaria, the A and B markers are not just attached to red cells. In many people, they also decorate the lining of the airways and the gut. The surfaces the virus meets first. That makes them potential docking points, or at least parts of the landscape the virus has to cross. One idea, borrowed from work on the original SARS virus, goes like this. When SARS-CoV-2 buds out of an infected person's cells, it carries with it a residue of that person's blood group sugars, like a coat borrowed from its last host. If that host is group A, the viral particles may be dusted with A-like decorations. Now imagine those A-coated viruses moving into a group O or group B person, someone whose immune system naturally makes anti-A antibodies. In theory, those antibodies could stick to the incoming virions and interfere with their ability to bind to ACE2 receptors, the doors the virus uses to enter cells. The result would not be absolute protection, but a slightly more hostile environment for viral entry. A person with group A, by contrast, does not make strong anti-A antibodies. If they inhale a cloud of A-decorated virions from another A-type, nothing in their plasma recognizes that sugar as foreign. The virus meets less immediate resistance. This scenario is elegant. It fits with the old SARS-CoV-1 data. It also remains, for now, partly hypothetical. Demonstrating it in the messy reality of living lungs and real-world exposures is much harder than sketching it on paper. Another, more grounded mechanism comes from hematology, a field that has been measuring the side effects of ABO types for decades. Long before COVID-19, researchers knew that people with non-O blood, A, B, and especially AB, tend to have higher baseline levels of certain clotting factors, particularly von Willebrand factor and factor VIII. That means, on average, a slightly higher tendency toward blood clots, the microscopic nets of fibrin and platelets that can plug a wound, or a vessel. When severe COVID-19 cases began to reveal strange patterns of clotting, tiny clots in the lungs, strokes and heart attacks in relatively young patients, hematologists were not entirely surprised to see non-O groups, especially A, showing up more frequently among those with the worst thrombotic complications. Again, this does not mean that having A or AB blood dooms you to a clot if you catch the virus. It means that, in the complex tangle of inflammation, endothelial damage, and immune overreaction that defines severe COVID, starting with a higher baseline of clotting factors might tilt the scale a little. A B in this light becomes particularly intriguing. As the double non-O type, AB often sits at the high end of von Willebrand factor and factor VIII measurements. In theory, that could make AB individuals a bit more prone to the clotting side of severe COVID. Some studies did report slightly elevated risks in AB carriers, others did not. With AB making up only a small fraction of any given population, its signal was often drowned by small sample sizes and confounding variables. Still, to immunologists and geneticists, AB's position is irresistible. It is the only common phenotype that displays both A and B antigens, and makes neither anti-A nor anti-B antibodies. It lives at a crossroads of tolerance and recognition, an outlier in how the immune system learns what to ignore and what to attack. Studying how AB people responded to SARS-CoV-2, how their antibodies developed, how their clotting profiles changed, offered a natural experiment in what it means, in the 21st century, to carry the fullest version of the ABO code. When you put all the COVID and blood type research together, one theme keeps emerging: subtlety. Yes, there are hints that ABO status can nudge your risk of infection by a few % one way or the other, and that it might colour the way your blood vessels and platelets behave under viral stress. But these nudges are tiny compared to the heavy hands of age, chronic disease, and access to care. A vaccinated 80-year-old with Group A is almost certainly safer than an unvaccinated 40-year-old with Group O. The virus does not read your ABO label first. It reads the state of your lungs, your immune memory, your social circumstances. In evolutionary time, though, those tiny nudges matter. Imagine a world without intensive care units or vaccines, where a respiratory pandemic sweeps through every decade or two. In such a world, if Group O truly does face slightly lower infection odds, and non-O types face a slightly higher risk of lethal clots, those edges could, over centuries, translate into small shifts in allele frequencies. Not dramatic sweeps, just quiet adjustments, one more layer in the long negotiation between pathogens and the ABO locus. But we do not live in that world anymore. Modern medicine, public health measures, and technology have wrapped most human populations in a loose net that catches a great many who would once have been lost. Antibiotics, oxygen, anticoagulants, steroids, antivirals. These interventions blur the fine lines that ancient infections could draw so sharply. COVID-19 reminded us that the old dance is still going on beneath that net. The virus probed the same old structures, the same sugars and clotting pathways that cholera and malaria and smallpox had pressed against before. Our blood types responded with the same minuscule shifts in odds. But for the first time in our species' history, those genetic whispers competed not just with other genes, but with ventilators, mRNA vaccines, and global data sharing. AB, the rarest of the main groups, remained what it has always been, a minority strategy, balanced between risks and benefits that depend heavily on context. In some infections, its dual antigens may be a liability. In others, its lack of anti-A and anti-B antibodies may soften harmful immune reactions. COVID-19 did not settle the question. It simply added one more data point to a story that stretches back through cholera rivers and malarial swamps and plague-stricken cities. When you lie in bed and feel your pulse beating quietly in your wrist, you're feeling the sum of all those negotiations. The virus that circled the globe in 2020 is just the latest in a long line of microbial diplomats testing the treaties written into your cells, and the letters A and B sitting together on the red cells of an AB person are not just curiosities on a transfusion card. They are signatures on a contract, a record of deals struck between your ancestors and their invisible enemies. How we choose to read those signatures is another story, because once humans knew that blood could be sorted into types, they did not stop at medicine. They carried the categories into culture, identity, even morality. And the small, strange minority who carry AB found themselves given meanings far beyond molecules. The first time someone tried to map races with blood, it was on a battlefield. Not in the heat of fighting, but in the long, uneasy quiet afterward. World War I had thrown men from every corner of Europe and its empires into the same muddy trenches, the same field hospitals, English and French, Germans and Austrians, Poles and Serbs, Indians, North Africans, Turks. Bodies that would never otherwise have shared a campfire now bled on the same stretch of earth. For Ludwig and Hanka Hirschfeld, a Polish-Jewish husband and wife team working as medical scientists on the Macedonian front, this chaos was an opportunity. By 1919, Landsteiner's A, B and O were no longer mysteries. They were tools. The Hirschfelds began quietly collecting samples and recording the blood types of thousands of soldiers, more than 8,000 in all, from at least 16 nationalities. Then they did something new. They didn't just note these blood groups for transfusion safety. They counted them by origin, by language, by the rough categories people called race and nation. English soldiers, many more A than B, Indians, less A, more B, French, Italians, Serbs, North Africans, each with its own pattern of A, B, and O. From these ratios, the Hirschfelds proposed what they called racial biochemical indices, numbers that, in their view, could help chart human groups the way a chemist charts elements on a table. They were not alone in thinking this way. The early 20th century was drenched in typologies, skull shapes, skin colours, Nordic, Mediterranean, Alpine, endless attempts to turn messy human variation into neat, bounded kinds. Now, for the first time, blood itself seemed to offer a secret code. Imagine the appeal, an invisible signature written inside the body that no one could fake or hide. Something more objective, more scientific than looks. From this came a new word, seroanthropology, the study of peoples through their sera, their blood. On paper, the idea had a quiet, almost clinical charm. Measure A and B frequencies here and there, draw contours on a map. In one region, A rises and B falls. In another, O dominates. Connect those curves and you get smooth gradients, clines, spreading like weather systems across continents. But the world that received these maps was not neutral. In the 1920s and 30s, as the Hirschfeld's tables and others like them circulated, German nationalists and eventually Nazi race scientists seized on them. The gentle clines of serology hardened into front lines in their imaginations. High A in Northern and Western Europe? That could be cast as Aryan. Higher B in parts of Asia, Eastern, Asiatic. Mixed patterns at borders, signs of unwanted blending. Never mind that every blood type appeared, in some proportion, in every population. Never mind that the very data they used showed enormous overlap, not clear separation. For ideologues looking for biological proof of hierarchy, ABO charts became convenient props. Within that framework, AB occupied an uneasy place. It was rare to begin with, typically a few % of any given population, higher in some East Asian groups, lower in many European or African ones, and it was by definition a combination, A and B together on the same cells, a genetic handshake between two ancient alleles. To a geneticist, this is straightforward codominance. To a eugenicist obsessed with purity, it could look like something else. A B could be framed as hybrid, mixed, a sign that different bloods had met. In an ideology that worshipped unmixed lines and feared degeneration, that was enough to make it suspect, even if most propaganda did not linger on the technical details. So the same molecules that kept transfusions safe were drafted into a very different project, justifying who belonged and who did not. In lecture halls and pamphlets, blood group frequencies were held up as hard evidence that races were real, fixed biological entities. The ABO system, discovered to explain why some patients lived through transfusions and others died, was now invoked to explain why some people should live and others could be sacrificed. At the same time, another story was quietly unfolding in courtrooms. Judges and lawyers had their own reasons to care about ABO. They were not drawing maps of races. They were deciding paternity suits and criminal cases. By the 1920s, doctors had noticed something useful. While blood type could not prove that a particular man was a father, it could often prove that he was not. Two Group O parents could not have a child who was AB. A man with Group B could not have fathered a child whose blood type required an A allele, if the mother had none to give. Soon, in Europe and the United States, mothers and alleged fathers were being ordered to roll up their sleeves. A B, with its dual antigens, became especially helpful for these exclusions. If a child was AB, at least one parent had to contribute an A allele and one a B. The mathematics of inheritance turned into legal arguments about responsibility and support. Forensics followed. Blood stains at crime scenes were typed as A, B, AB, or O. Again, this could not uniquely identify a person the way DNA profiling would decades later, but it could exclude suspects or narrow a list. In a world before genetic sequencing, ABO markers were one of the first biochemical tools that could link a body to a story in law. So, in the space of a few decades, blood groups had been enlisted in two opposing dramas. In one, they were instruments of exclusion, used to construct racial hierarchies and fantasies of purity. In the other, they were instruments of clarification, helping establish or deny biological ties between individuals, cutting through lies with simple Mendelian logic. Modern genetics would eventually step in as a kind of auditor, reviewing the books kept by those early serologists. When population geneticists took the same ABO frequency tables that race scientists had used and analyzed them with better statistical tools and a different set of questions, the picture changed. They found that most variation in these markers existed within populations, not between them. They saw that A, B, O, and AB did not arrange themselves in sharp blocks matching the old racial categories, but in smooth, overlapping gradients across geography. High B in some parts of Central Asia, yes, but trailing off gently into Europe and India. O common among indigenous peoples of the Americas, but also heavily represented in parts of Africa. Clines, not walls. The conclusion many drew was not that biology is irrelevant, but that race, as a small set of discrete, homogeneous blocks, does not match how human genes, including ABO, are actually distributed. Blood type became, ironically, one of the tools that undermined race as a strict biological category. A B, in this newer light, looks very different from the hybrid of eugenic imagination. It is still a minority strategy, still one of the rarest common types, but instead of standing for impurity, it stands for something universal, contact. To have a B blood is simply to carry both A and B alleles, the way millions of people do. It means somewhere in your family line, often in many places, those alleles met and combined, not once, but over and over, as people moved, married, had children across whatever social boundaries existed at the time. In that sense, AB is not an exception to human history. It is a visible emblem of its most ordinary feature, mixing. Every human population shows some evidence of admixture, of gene flow from neighbours or travellers, from conquerors and refugees, from people whose names are not in any surviving record. The ABO system records a tiny part of that blending. Where A and B both circulate, AB will appear, quietly, whenever two carriers meet. When you hear older language about pure blood and mixed blood, it is worth remembering this. No blood type belongs to a race. No letter on a transfusion card knows what passport you carry. The A allele found in a Norwegian patient and the A allele in a Kenyan nurse may be more closely related to each other than either is to some other A in their own country. The patterns we see, like slightly more B in some regions, more O in others, are traces of migrations, epidemics, and chance, not of fixed, separate kinds of humans. The abuse of blood science in the age of eugenics has left a long shadow. It is one reason many biologists today are careful, almost cautious, when talking about human differences. They know how easily numbers can be turned into weapons, how eagerly the human mind reaches for categories that feel absolute and moral. So when geneticists say that ABO variation is clinal rather than racial, they are not just making a technical point, they are offering a kind of antidote. They are reminding us that the gradients on the old serological maps do not mark natural castes, but the ebb and flow of people over time. They are saying that blood groups are part of our shared story with disease and movement, not a justification for ranking each other. And AB, the strange newcomer in the history of transfusion, becomes something more than a curiosity. It is a letter pair that once made race hygienists uneasy, a combination that did not fit their neat boxes. Today it can stand, if we let it, for the breakdown of those boxes, for the fact that human lines have always crossed, that no one's ancestry is simple or clean. The same antigens that once were used to sort and divide can just as easily be read as signatures of connection, from race hygiene pamphlets to paternity rulings, from trench hospitals to quiet genetic labs. The story of ABO and identity has been one long struggle over how to read what is written in our blood. In the next part of this story, those readings get stranger, because once the old racial hierarchies began to crumble, people did not stop assigning meanings to A, B, and AB. They simply moved the meanings elsewhere, into personality, into romance, into diet plans and pop psychology. A B, rare and liminal, would become, in those tales, not a suspect hybrid, but a mystical outlier. In Japan, this mysticism acquired a name, Ketsueki Gata, literally blood type form, or blood type style. A way of saying that the letters on your transfusion card were not just medical information, they were a kind of horoscope. The idea surfaced in 1927 in a short paper by a Japanese psychologist named Takeji Furukawa. He called it the study of temperament through blood type. At the time, Japan was wrestling with questions about education, discipline, and how to classify students. Furukawa suggested that the answers might lie under the skin. Type A, he wrote, tended to be earnest, anxious, orderly. Type B, individualistic and free-spirited, maybe a bit selfish. O, confident, outgoing, the natural leader. And AB, that was the puzzle piece. AB in Furukawa's early typology was the mixed type, rational but indecisive, creative but eccentric, cool on the surface but internally conflicted, a person of dual nature, combining A's seriousness with B's originality for better or worse. Other psychologists criticized the work. The data were thin, the statistics crude, but the categories escaped the journals. They drifted into magazines, casual conversation, schoolyard chatter. They stayed there. Then, in the 1970s, a journalist named Masahiko Nomi picked them up again and turned them into a publishing phenomenon. Nomi was not a laboratory scientist. He was a collector of stories. He gathered anecdotes about friends, celebrities, colleagues, and arranged them under Furukawa's headings. The result was a series of bestsellers claiming that you could understand yourself and others through blood type. Once again, AB took on the role of the rare, mysterious outlier. Television shows and women's magazines described the B-types as genius or oddball, logical in one moment and dreamy the next. The person who sits on the edge of the group, watching, thinking, then suddenly offering an insight no one else had noticed. In a country where only a small minority has AB blood, that image stuck. By the early 2000s, blood-type personality had become a quiet background hum in Japanese daily life. Dating profiles listed not only height, hobbies and favourite foods, but also A, B, O or AB. Celebrity bios on Variety shows flashed blood type beside age and birthplace, as if it were another basic fact. Morning talk programs devoted segments to today's blood type fortunes. Shelves in bookstores sagged under How to Get Along with an A-Man and Raising Your B-Type Child guides. In South Korea, Similar ideas caught on. Matchmaking agencies asked for blood type. Pop idols' profiles included it. Fans debated whether their favourite singer's onstage persona matched the official stereotype. Sometimes the beliefs had a playful, almost cosy flavour, like light astrology. Friends teasing each other, "Of course you're late, you're B," or "You're so calm, you must be O." A B in these conversations became the enigmatic friend, the one people describe with a mix of affection and bafflement. But there was a sharper edge too. When people start to treat any typology as real, they can start to treat it as fate. In Japan, a new word appeared: burahara, blood harassment. Stories circulated of bosses who preferred A-types for accounting and O-types for sales, of co-workers blaming team conflicts on a colleague's supposedly difficult B-blood, of parents worrying that their AB child would struggle to fit in. There were reports, even if not universal practice, of schools casually grouping kindergartners by blood type to balance personalities. None of this was enforced by law. There were no official decrees. But the stereotypes seeped into expectations, and expectations have a way of shaping lives. Underneath it all, one simple question lingered: is any of it true? Psychologists tried to find out. In Japan, Korea, and elsewhere, researchers gave thousands of people personality tests, then checked their ABO types. Over and over, the same pattern appeared. When you measure traits carefully, introversion, conscientiousness, openness, neuroticism, and compare them to blood groups, the differences are tiny to non-existent. Some studies pick up slivers of correlation, but they are weak, often explained by people answering in ways that match what they've been told about their type. If you believe AB people are eccentric and indecisive, and you know you are AB, you may rate yourself slightly more that way. Not because your red cells are telling your brain what to do, but because culture is. When statisticians step back and look at the whole picture, the conclusion is consistent. There is no robust evidence that having A, B, O, or AB antigens on your cells causes you to be tidy or messy, shy or bold, romantic or cool. Blood-type personality theory sits, scientifically speaking, in the realm of superstition. And yet, it doesn't feel like pure nonsense to many people who use it. There's a reason. Human beings love simple categories. We live in a world of overwhelming complexity, millions of individual quirks, histories, experiences. Systems like Ketsueki Gata compress that noise into 4 tidy boxes. You can watch the mind relax when it encounters a story like that. Ah, that's why I am like this. That's why he doesn't understand me. That's why we get along. Psychologists have a term for part of this effect, the Barnum effect. Give people a vague, flattering description. You are sometimes sociable, sometimes reserved. You value honesty but sometimes doubt yourself, and most will recognize themselves in it. Blood type descriptions are often like that, broad enough to fit almost anyone, specific enough to feel personal. A B, framed as rational but indecisive, creative but eccentric, is a perfect Barnum sentence. Who hasn't felt rational in one moment, indecisive in another? Who doesn't sometimes think of themselves as a little different? So the myth persists, not because red cells through some unknown pathway are sculpting personality, but because the story is satisfying. A similar hunger for bodily explanations underlies another modern AB myth, the blood type diet. In 1996, a naturopathic doctor named Peter D'Adamo published a book with a seductive title, Eat Right for Your Type. The premise was simple and sweeping. Each ABO group, he argued, reflects an ancestral lifestyle shaped by evolution. Type O was the hunter, suited to high-protein meat diets. Type A, the farmer, better with grains and vegetables. Type B, the nomad, tolerant of dairy. And AB, the youngest type, a modern blend that should eat a mixed A/B diet, largely plant-forward, like A, but with some of B's dairy and flexibility. He went further, proposing that certain lectins, proteins in foods, would react badly with particular blood types, agglutinating their cells or causing subtle immune problems. Match your diet to your type, he suggested, and you could lose weight, reduce disease risk, feel better. Ignore it, and you might be quietly sabotaging your health. The book sold millions of copies. It spawned recipe guides, spin-offs, online quizzes. For AB readers, it offered something particularly alluring, a curated menu for a rare, sensitive bloodline. Later, scientists asked the same thing they had of Quetzalcoatl. Does it hold up? Nutrition researchers look for clinical trials, controlled studies, any solid evidence that tailoring diet to ABO type made a difference beyond general healthy eating. They did not find much. A systematic review published in a major nutrition journal in 2013 examined the available data and concluded that there was no good evidence to support blood-type diets. Later observational studies found that people who ate, say, a plant-heavy diet resembling the type A plan often did show better heart and metabolic markers, but this was true regardless of whether their actual blood type was AOB or AB. In other words, the benefits followed the food, not the letters. The proposed mechanism, lectins binding differently to different blood groups in the living human body, also looked shaky. Most lectins are broken down in cooking and digestion. The dramatic reactions seen in test tubes do not map neatly onto what happens in a stomach or intestine. Professional organizations and evidence-based summaries today are blunt. There is no strong scientific basis for eating by ABO type. And yet, like personality typing, the blood-type diet has an intuitive pull. The idea that your unique blood chemistry could tell you what to eat feels modern and old at once. Part genetic testing, part folk wisdom. It offers a map through the anxiety-inducing landscape of nutritional advice. It says, you don't have to make endless choices. Your body has already chosen for you. For someone with AB blood, rare and often labelled special, a personalised list of recommended and forbidden foods can feel almost like a secret manual. When we step back from these myths, Quetzalcoatl, blood-type diets, we can see a pattern. None of them are supported in their strong forms by good biological evidence. Large studies do not find that AB people share a distinct blood-driven personality profile. Controlled trials do not show that AB-specific diets have unique effects on AB bodies, but the myths themselves are not meaningless. They tell us what people want from biology. They show a deep desire to anchor identity, our tendencies, our loves, our health, in something tangible and inherited, to read the self in the body, to find order in the great tangle of temperament and appetite. In that sense, the stories are not about AB at all. They're about us, looking for ourselves in whatever mirror science happens to offer. Sometimes that mirror is clear, as with transfusion compatibility or disease risk. Sometimes it is fogged by wishful thinking and cultural fashion. AB, being rare and inherently a combination, becomes an especially attractive screen for projection. Mysterious, liminal, something apart. The perfect candidate for roles like genius, oddball, modern hybrid, sensitive type. But behind the projections, the biology remains stubbornly simple. A B is a codominant genotype at a single locus, inherited by the same Mendelian rules as A, B, and O. Its antigens matter for transfusion and for a handful of infections. Beyond that, your thoughts, feelings, and best dinner choices are shaped far more by the intricate interplay of thousands of genes, environment, culture, and chance than by this one set of sugars on your red cells. If anything, the persistence of AB myths is a quiet reminder to handle all biological categories carefully. They can help us stay alive in operating rooms, They can help us trace ancient migrations and understand past plagues, but they can also tempt us into seeing patterns that are not there, into sorting ourselves and others into boxes that feel profound but rest on very thin ice. And yet, even these misreadings are part of the story our blood carries. From Furukawa's classrooms to Nomi's paperbacks, from Tokyo talk shows to diet guides on foreign shelves, AB has been drafted again and again into narratives about what kind of person you're allowed to be. In the next turn of this story, it will leave the realm of fortune-telling and food lists and return to the quiet world of hospitals and blood banks. Because whatever myths have grown up around it, Type AB has a very real, very practical role to play there. One that depends not on who you are, but on what your plasma can do for someone you will never meet. If the world of Ketsueki Gata is all about what your blood is supposed to say about you, the world we're about to enter is the opposite. Here the letters don't describe personality, they decide who lives through the night. Imagine a different kind of network now. Not bands of hunter-gatherers scattered across ancient Africa, but hospitals scattered across a continent. Islands of light in the dark, each one surrounded by highways and neighborhoods instead of savanna and scrub. In their basements and side wings, behind locked doors and humming refrigerators, there is another kind of meta-population. Bags of blood, packets of platelets, frozen blocks of plasma. Constantly arriving, quietly expiring, always on the move. In that network, AB is tiny. Only about 3 or 4 people out of every 100 who roll up their sleeves to donate belong to the AB group at all. AB negative, with its extra twist of RH status, is rarer still, hovering around 1 person in 100 in places like the United States and United Kingdom. In some communities it's even less. On paper it looks like an evolutionary afterthought, a marginal phenotype, occupying the far corner of a compatibility chart. But in the blood bank, rarity and importance do not always move in the same direction. To see why, you have to turn the ABO story inside out. Earlier, we followed the antigens, the A and B sugars perched on red blood cells, like flags announcing identity. In transfusion medicine, another character steps forward, the antibodies floating in the plasma that surrounds those cells. Most people, somewhere in the first months of life, quietly start making antibodies against whichever ABO antigens they do not have. Type A people make anti-B. Type B people make anti-A. Type O people make both, anti-A and anti-B. These antibodies are vigilant. If they meet red cells carrying the wrong flag, they grab them, clump them, and call in the immune system's cleanup crews. It's an efficient defense mechanism against certain kinds of invaders. It is also the reason mismatched transfusions can be fatal. But AB people are different. Because their own cells carry both A and B antigens, their immune systems learn early on that both are self. They do not make anti-A or anti-B. Their plasma is, from the ABO perspective, disarmingly tolerant. If you are lying on an operating table, bleeding fast, and the surgeons do not yet know what your blood type is, this detail matters. You cannot safely receive any random red cells. That would be like playing Russian roulette with your circulation. But plasma, the pale straw-coloured liquid that carries clotting factors, hormones, and those all-important antibodies, can in some situations be poured in before the exact typing results are back. If that emergency plasma comes from a type O person, it will almost certainly be loaded with both anti-A and anti-B. Good for them, dangerous for you, whoever you are. If it comes from an AB donor, it will contain neither. That absence is almost magical in its simplicity. A B plasma does not come looking for A or B to attack, because it has been trained its whole life to accept both. This is why transfusion services often call AB plasma universal. In practice, with careful cross-matching and awareness of other blood group systems, it can be given to patients of any ABO type. In trauma bays and burn units, in neonatal intensive care, In military field hospitals, this makes AB plasma an emergency tool of choice. When time is measured in heartbeats, the ability to hang a bag that will not pick a fight with whatever red cells a patient has is priceless. And yet, remember the numbers. Only a thin sliver of the population can supply this universal fluid. Both AB positive and AB negative plasma have this special property, but AB negative donors sit at the very edge of rarity and utility, the smallest group whose donations often have the widest safe reach. It's an odd inversion. On the red cell side, AB people are the opposite of universal donors. Their red cells carry every ABO flag. AB positive cells carry every ABO flag and the RHD antigen as well. You cannot casually drip those into the veins of an unsuspecting stranger. Almost everyone will have antibodies against something on their surface. AB individuals are, in the old teaching phrase, universal recipients of red cells. If you are AB positive, your immune system has already made peace with A, with B, and with RHD. You can safely accept red cells from almost anyone, A, B, AB or O, positive or negative, as long as other, more obscure blood group systems are compatible. It sounds like a selfish position in the exchange, a taker, not a giver. But if you shift your focus from the cells to the liquid, the picture flips. A B people become universal givers of plasma, quiet specialists whose donations are less about their own safety and more about everyone else's. In modern blood banking, this shapes an entire pattern of logistics. When a person with AB blood walks into a donation centre, staff often steer them gently toward a machine that does something more elaborate than a simple whole blood draw. It's called apheresis. The machine takes blood out, separates it into components with a spinning centrifuge, red cells here, platelets there, plasma over there, keeps the pieces the hospital most needs, and returns the rest to the donor's arm. The process takes longer than a standard donation, but it can yield more of the rare components in a single visit. For AB donors, the emphasized components are almost always plasma and platelets. Platelets are tiny, disc-shaped fragments that help blood clot. They're essential when someone's bone marrow has been suppressed by chemotherapy, when a person has bled heavily, when certain diseases have shredded the body's normal clotting capacity. Like plasma, platelets are suspended in fluid that carries antibodies. AB platelets, lacking anti-A and anti-B, can often be transfused more broadly across ABO lines than platelets from other types. Hospitals lean on them for complex cases, mismatched transplants, and situations where ideal ABO matching is hard to achieve in time. There's a catch, though, built into the physics and chemistry of the components themselves. Red cells, dense, iron-rich, store well in the cold. Under standard conditions, they can sit refrigerated for about 5 to 6 weeks and still do their job when transfused. Plasma, once frozen solid, is patient. It can hold in that state for many months, sometimes up to a year, waiting quietly for the call from an emergency room. Platelets are the opposite. They prefer room temperature and constant gentle agitation, conditions that keep them metabolically active. The trade-off is a shelf life measured in days, not weeks, 5 to 7 days typically, before they age out of safe use. The result is a continual race against the calendar. Blood banks can build up some reserve of AB plasma in freezers, creating a buffer for large-scale emergencies, but for platelets, stockpiles are flimsy. Inventories rise and fall quickly. A handful of trauma cases, a run of complicated surgeries, a bad week in a neonatal unit, and the shelves can thin. So the network adapts. Regional transfusion services keep lists, sometimes literal, sometimes in databases, of AB donors, especially AB negative. They call, text, send reminders. Not in a panic, but with a steady sense of urgency. Can you come in this week? We could use your platelets. We could use your plasma. Campaigns go out. were especially seeking AB donors. Posters and clinics mention, almost apologetically, that while every type is needed, some are needed in a different way. The rarest common type becomes a managed resource, a quiet priority in scheduling algorithms and phone trees. Globally, this pattern repeats with local variations. In countries where AB is a little more frequent, parts of East Asia for example, there is slightly more breathing room. In regions where AB is scarce or where donor pools are small for other reasons, AB-negative plasma and platelets are treated almost like strategic materials. Hospitals trade components across city lines. National blood services shift frozen plasma inventories from one region to another ahead of predicted surges. Festival seasons with more traffic accidents. Winter stretches with more complicated medical admissions. The scale is enormous, but the basic principle is the same one that governed those scattered human bands after the ancient bottleneck. Redundancy, diversity. A network robust enough that when one node runs dry, another can help. A bee, tiny in numbers, sits at important junctions in that network. It is an irony with a certain narrative symmetry. The newest blood type in the scientific story, the one that puzzled early immunologists, the marginal phenotype that seems like an improbable hybrid, is, in the practical world of modern medicine, one of the most universally useful. Not because it can give its red cells to everyone, it cannot, but because, stripped of attack antibodies, its plasma can flow into almost any body without starting a war, because its platelets can patch leaks in veins and arteries without picking fights over A and B. Most of the time, this happens without anyone thinking of it as remarkable. A patient on an operating table does not know that the straw-coloured bag dripping into their vein came from one of the rarest donors on the chart. The surgical team is focused on sutures and blood pressure, not on global allele frequencies. The person whose arm fed the apheresis machine will walk out of the centre, drink some juice and go back to their day. Somewhere in that unbroken chain between donor and recipient, the letters A and B will be recorded. But they will sit there quietly, like a footnote. A B, in this setting, does not announce itself as special. It does not carry myths about genius or indecision. It simply does its job, over and over, behind the scenes, a late-arriving bloodline that, by a quirk of immunology, turned out to be exactly what high-tech medicine needed. A tolerant plasma, a flexible platelet, a rare resource that can move through almost any vein. A quiet hero in a world of refrigerators and fluorescent lights, where survival still depends, at the most basic level, on whether a few drops of someone else's blood can safely become part of your own. But the usefulness of AB doesn't end when the drip is closed and the bandage goes on. Once you've seen how a few milliliters of plasma can decide what kind of surgery someone survives, it's tempting to stop there. To think of blood type as a purely hospital-bound category, a label on a bag, a line in a chart, a compatibility rule memorized by transfusion nurses. Step back a little, though, and the letters start to do something stranger. They begin to act like clues, not clues that can tell you who you are or what kind of person you'll be, but clues that whisper about where your ancestors have walked, who they met along the way, and which invisible enemies they faced. If you could float up high above the earth, not into space, but just far enough that continents became patterns, you could imagine painting those patterns with ABO colours. Large stretches of the world would be washed in O. O dominates in the Americas, in much of Africa, across broad swathes of Europe. A great pale background, like the common ancestor from which other shades emerged. Then there would be regions where A is unusually strong, parts of northern and central Europe, pockets of Australia's settler populations, certain groups in East Africa. Elsewhere, islands and corridors of B would glow more brightly, across much of South Asia, through Central Asia's old caravan routes, in parts of East Asia and North Africa. These are not sharp borders, not lines on a map. They blur and overlap, shaped by migration, conquest, trade, and chance. But when you look at them together, something becomes clear. AB shows up most where those patterns meet. You rarely find AB dominating anywhere. Globally, it sits somewhere around 4 or 5% of people on average, rising in some places, sinking in others. In many Western populations, it hovers at 3 or 4%. In some crossroads, regions of North Africa and the Middle East, surveys have found AB edging up close to 10%. Those are not numbers that can tell you your ethnicity from a drop of blood. ABO frequencies can arise in similar ways in very different places. Drift and migration scramble the picture. Geneticists treat ABO more like a very rough regional accent than a precise address. But they can still read something from it at the level of populations. High B, low A, that might hint at old expansions from Central or South Asia. High A, low B, perhaps layers of European or certain African histories. A noticeable bump in AB sitting at the crossing point between A-rich and B-rich groups. That often marks a place where people with different blood histories have been meeting, marrying and having children for a long time. AB is, in that sense, the phenotype of a borderland. Not a border in the political sense, but a long-term frontier where two different ABO pasts have overlapped and had time to mix. Ports, trade routes, river deltas, mountain passes, places where caravans met, where empires rose and fell, where refugees moved through and sometimes stayed. Your own AB blood, if you have it, cannot tell you which of those stories is yours, but it does tell you that somewhere not too many generations back, the lines carrying A and the lines carrying B crossed in your family tree. That intersection is one kind of memory. For a long time, scientists tried to make those memories do more work than they really could. Before DNA profiling came along in the late 20th century, ABO typing was one of the few genetic tools available to courts and detectives. It was simple, cheap, and robust enough to survive outside a lab. You could dry a blood stain on a cloth, find it days later, and still ask, does this stain carry a antigen, B antigen, both, or neither? You could swab a suspect, type their blood, and see whether the categories matched. In murder cases, in assault investigations, in wartime inquiries, this was revolutionary. It didn't start with television-ready drama. It started with basic inheritance tables. By about 1910, researchers like von Dungen and Hirschfeld had shown that the ABO system followed Mendelian rules. A and B were dominant, O was recessive. This meant certain parent-child combinations were impossible. Two O parents could not, under normal circumstances, produce an A, B, or AB child. An AB parent and an AB parent could never have an O child. An O parent and an AB parent could not have an AB baby. From this, an entire early science of paternity and kinship testing sprouted. Hospitals and courts would line up the blood types of alleged fathers, mothers and children and look for contradictions. If the child's type was impossible given the supposed parents, that relationship could be excluded. Not proven false in an absolute sense. Rare mutations and recording errors always hover at the edges, but strongly, statistically unlikely. A B blood with its dual antigens became a useful part of that logic tree. If a child was AB and neither parent had A or B, something was wrong with the story. The same exclusion logic carried into forensics. A crime scene stain typed as AB could immediately rule out any suspect with type O, A or B alone. If a victim was type O and there was AB blood present, investigators knew there had to be another person involved. In some mid-century cases, that information helped narrow suspect pools before more detailed evidence was available, but there were hard limits baked into the math. A BO is a tiny fraction of the genome. Even when combined with a few other systems, RH, MNS, KEL, Duffy, KID, it could only ever push probabilities so far. Blood type evidence could say, this person is extremely unlikely to be the father, or this suspect cannot be the source of this stain. It could rarely say, this person must be the one. Then DNA arrived and the landscape flipped.